Hi, so I'm Jenna Galloway, and so I work on musculoskeletal regenerative biology. So how many people here have had some sort of musculoskeletal or orthopedic injury, anything from a sprain to strain to a tear? Yeah, so I mean, that's basically what my first slide is showing, is how common this really is. And it's not only something that affects the people as they get older, but it's also very common for young active individuals. It's actually, musculoskeletal injuries are among the most highly reported medical condition for people under the age of 50. So it sort of spans the entire population. In my lab in particular, we're focused on understanding the biology behind tendons and ligaments to be able to harness that for regenerative medicine applications. And we're interested in this in particular because not only is it a relatively understudied component of the system, people have studied a lot of the muscle, cartilage, bone, but also because it, again, affects a large percentage of the population. So I'm just showing some examples up here, such as torn rotator cuffs. So they average people over 60, approximately 50% of the population has a tear in their rotator cuff. And other incidents of tears, such as with the ACL or Achilles tendon, also results in several 100,000 of operations per year. And our current limitations, or the current treatments for these, are really surgery. They reattach the tear, try to have it heal over a time course. They also can use tissue grafts, so autologous autografts, allografts, and xenografts. So this is what they use in ACL tears to try and replace the structure. And currently there's some new materials out there called orthobiologics, where they're using manufactured tissue matrices to kind of bridge the site. But with all of these applications or current treatments, we still have a very slow and limited healing. So you can see the timeline that I put up here, where you have generally a month or a couple months of inactivity or immobilization at the injury site, followed by a physical therapy regimen with a slow return to activity, and can take 6 months, sometimes a year, to get back to what you were originally doing. And even with that, a lot of people are even left with pain, limited mobility, or this they're setting up for either having osteoarthritis later in life or a repeated tear or injury to the site. There are some current experimental strategies out there that are being used, and a lot of this ends up in the news media: platelet-rich therapy, stem cell therapies. But currently, the orthopedic major associations have kind of pegged these as very experimental. And there are some promising results, although it's thought to be more in the sense of their ability to regulate the immunomodulate the healing, so that you decrease the inflammation at the site and they're not actually contributing to the repair. But one thing that I think can strike you looking at the current treatment and approaches is there are currently no drugs for tendon and ligament injuries that are out there on the market. And that's where my lab has been working towards, is trying to engineer a platform that we can use to identify drugs that can be used in regenerative therapies to, as we've been hearing from the other speakers, to stimulate the endogenous cells to repair the tissue, or to use them in a tissue replacement strategy approach. And the way we're doing this is using the zebrafish, which Mark introduced a little while ago. And so these are organisms with very high gene homology and pathway homology to humans. And so we're using the zebrafish system that has its marked, its cartilage and tendon tissues. And what we're looking for are drugs that promote the formation of these tissues. And so we did what the zebrafish enabled us to do in a very rapid and cost-effective manner, was to identify a cohort of drugs from known biochemical and FDA-approved libraries that promote tendon and cartilage fates. And what we're currently in the process of doing is now testing their activity in human cells using different stem cell assays. And we're finding drugs that have conserved activity, being able to promote these particular lineages, with the idea being They could be used for promoting these fates in culture to make tissue replacement parts, so far for instead of using autologous transplants, or as actual therapeutics, where you could inject these drugs or provide a matrix that is enriched with these drugs to stimulate the tissue to better repair. And so that's what we think the future of this particular structure or of your musculoskeletal system is. So thank you very much.